and welcome to Miss Estruch Biology and this entire Topic 5 theory video for AQA A-level Biology. If you want help making notes as you go, then don't forget to check out my free workbook that you can download listed in the description. But if you want even more help, for example, lessons with me plus multiple lessons per week with examiners and teachers and some of my resources for free, plus a study chat group, plus mock papers that I mark and so much more, then don't forget to check out the Biology Study Club, which is the monthly membership club that I run. I'll link it in the description. But that's it for now. Enjoy the video. AQA Topic 5. And we begin with 3.5.1 photosynthesis. And everything in Topic 5 is A-level only, not for AS. Now, the first part of photosynthesis is a little recap on the structure of a chloroplast because this is where photosynthesis happens. You have a double membrane organelle, and then the thylakoids are folded membranes which contain photosynthetic pigments such as chlorophyll. And these are going to be involved in the light-dependent reactions in photosynthesis. You also have this fluid-filled centre called the stroma, and that is where the light-independent reactions happen. Now, chlorophyll is actually made up of many photosynthetic pigments, and these are in those proteins embedded within the thylakoid membrane. And the advantage of there being multiple photosynthetic pigments is each of those pigments absorbs light at a different wavelength. And that then means there's a bigger spectrum of visible light that is able to be absorbed by the plant. And if a bigger spectrum of wavelengths can be absorbed, that means overall more light energy is absorbed. And if more light energy is absorbed, then there's more energy for the light-dependent reactions. So the light-dependent reactions then, this is the first stage in photosynthesis, and it happens on the thylakoid membranes, or a stack of them is known as a granum. The light-dependent reactions require light energy and they use water, and they produce ATP and reduced NADP. And that is through these three stages: photolysis of water, photoionization of chlorophyll, and chemiosmosis. So we're going to go through those three and see how it results in the production of ATP and reduced NADP, which are needed for the light-independent reactions. Photolysis of water, photo, if you see that in biology, it means light, lysis means splitting. So it is using light energy to split water. That is what photolysis is. And we've got an equation here. Water, and it's using the light energy, is split into oxygen, electrons, and protons, also known as hydrogen ions. The protons are going to be used to reduce NADP, and that helps to maintain a proton gradient in one of the later stages. Electrons, those are going to replace the electrons that get lost in chlorophyll in one of the stages we look at. Oxygen, this is a byproduct, it's not actually used. So a few things just to point out here. NADP is a coenzyme. These are not enzymes. They don't catalyze reactions. They are going to be carrying molecules from one point of a reaction to be used later. And if it's reduced, it means it's gained a proton and an electron. So that is what we mean by that term. Photoionization of chlorophyll. This is when light energy, again, so photo meaning light energy is absorbed by the chlorophyll, and that energy causes an electron to be released. So it's been ionized. But they are really specific with the terminology you use for this. So you'd have to say the light energy is absorbed by the chlorophyll. That's usually 1 mark. Then it's saying what that light energy does. So it results in the electrons becoming excited and raising up an energy level. That is the language they put in the mark scheme: excited and raising up an energy level. And that causes the electrons to be released, and therefore chlorophyll has been ionized. And those electrons are going to be used in one of the later stages to help make ATP and reduced NADP. And that later stage I keep referring to is chemiosmosis. This is where we're going to see the protons and electrons being used to make ATP and to make reduced NADP. NADPH is the same thing as reduced NADP. Either of those will be accepted on the mark scheme. So step 1 Here is PS2. You don't need to know that terminology for AQA. That is just a protein embedded within the thylakoid membrane. And we can see here, this is the thylakoid lumen. This is the chloroplast stroma. So within the thylakoid lumen, so within inside of the thylakoids, that's where we have our photolysis happening. Water has been split into protons and electrons, which are picked up by this protein, and the oxygen's a byproduct. So we can see there we've got our electrons, and those are replacing the electrons that are getting excited, raising up an energy level, and leaving the chlorophyll, moving on to the next protein in the photoionization of chlorophyll. So we get these electrons moving along this chain of proteins, and every time they move from protein to protein, they release energy. And that energy is used to pump protons from the stroma into the thylakoid lumen. And as a result, from those protons being pumped across and the protons released from photolysis of water, we end up with lots of protons on the thylakoid lumen side compared to the stroma, which means we've created an electrochemical gradient. Those protons can move down that electrochemical gradient back into the stroma, but it has to be through a protein, and the only protein they can move through is the enzyme embedded within the thylakoid membrane, ATP synthase. So the protons move through ATP synthase, and that enables that enzyme to catalyze the phosphorylation of ADP into ATP. So that is how ATP is made in chemiosmosis. The last step then is, how is this reduced NADP made, or NADPH? Well, the protons that have moved through ATP synthase are picked up by that coenzyme NADP, and also the electrons, when they get to the end of this electron transport chain, they are also picked up by NADP. And that then forms reduced NADP, or NADPH. And that's how we get those two products that we're now going to see used in the light-independent reactions, also known as the Calvin cycle. This stage happens in the stroma. It uses the enzyme Ribisco, so it's temperature sensitive. So the key things you need to know about the Calvin cycle are, it's going to use carbon dioxide. It also uses reduced NADP and ATP. The ATP is going to be hydrolyzed to provide energy for reaction. The reduced NADP is going to donate hydrogen, which is a proton, and an electron, and therefore it will reduce molecules in the cycle. We're going to have a look at this whole cycle, but that's just a summary to be aware of. So here is the cycle. Step one is carbon dioxide is going to enter the cycle, and it combines with RUBP, which is a 5-carbon compound. So a 5-carbon compound, RUBP, is going to combine with carbon dioxide, a 1-carbon compound, and that is catalyzed by Ribisco. So that adds up to 6 carbons, but we actually get that splits to make 2 3-carbon compounds known as GP. So all of that is step number 1. Those 2 molecules of GP are then reduced to form 2 molecules of triose phosphate. So the way that they are reduced is using the reduced NADP. So the hydrogen from reduced NADP is gained by the GP molecules to reduce it. The ATP is hydrolyzed to provide the energy for that reduction. Now, some of those carbons from the triose phosphate are going to be removed each turn of the cycle. And it's actually going to be 1 that is removed. And that means if we're removing 1 carbon each time, we need to make a hexose sugar, such as glucose, the cycle has to happen 6 times in order for those useful organic substances, such as hexose sugars, to be made. But each turn of the cycle, because 1 carbon is removed, that leaves 5 carbons remaining. And it'd be as a 3 and a 2. And we need to join those back together to regenerate RUBP. And that's why ATP is also needed at this stage in the cycle. ATP is hydrolyzed to provide the energy to regenerate RUBP. Now, whilst glucose is the product, generally speaking in the mark scheme, they'll accept you saying organic substances, meaning it contains carbon and hexose sugars. But you can also get questions about what could that go on to form, linking to your knowledge of biological molecules. So an organic molecule contains carbon, so you could be creating glucose, sucrose. Those could then go through condensation reactions to make polysaccharides such as cellulose or starch, but it could also be converted into glycerol, and then the glycerol can combine with fatty acids to make lipids, or it could be used to make an amino acid if you add an amine group. There are limiting factors that reduce the rate of photosynthesis. Those are the light intensity, carbon dioxide concentration, and temperature. The light intensity is going to limit the rate because it provides the energy in the light-dependent reactions. So if you don't have a high enough light intensity, then photolysis of water and photoionization of chlorophyll won't be happening at a fast rate. However, it will get to the point where it can get brighter and brighter and the rate still remains the same because maybe you now have a different limiting factor such as carbon dioxide concentration or temperature. Carbon dioxide concentration can limit the rate because it is the reactant in the Calvin cycle. Again, it will plateau eventually because maybe you don't have a high enough light intensity or the optimum temperature. Temperature is linked to the idea of it being an enzyme-controlled reaction. So at too low a temperature, there's not enough kinetic energy for successful collisions, and therefore you don't get as many enzyme-substrate complexes. And too high a temperature, the enzyme denatures, active sites change shape, so again you don't get as many enzyme-substrate complexes. Now, for maximum photosynthesis, and therefore plant growth, in farming and agricultural practices, they want to be removing limiting factors so they get more crop to sell. And that could be by increasing the light intensity, the temperature, and burning fuel to release more carbon dioxide. But all of those strategies cost money. So they will have to work out if the extra growth from photosynthesis offsets the cost of paying for extra heating, lighting, and fuel. And this often comes up as application questions linked to experiments where you have to work that out. Next is 3.5.2, respiration. There's aerobic and anaerobic respiration, and we are going to start with aerobic, which is split into 4 key stages. We have glycolysis, which happens in the cytoplasm of the cell, but the other 3 stages do happen in the mitochondria. The link reaction and the Krebs cycle happen in the mitochondrial matrix, which is the fluid-filled center. And oxidative phosphorylation happens on the cristae, or the inner mitochondrial membrane. And this is an enzyme-controlled reaction, so it is sensitive to temperature changes and pH changes. The respiratory substrate typically is glucose, and that is broken down through the reactions and it will release ATP. But if there's no glucose, then glycogen in animals could be hydrolyzed into glucose, or if it's a plant, then the starch could be hydrolyzed into glucose. Now, if there's no glucose or glycogen in animals or starch in plants, then the next option is other biological molecules will need to be used. So lipids can be hydrolyzed into glycerol and fatty acids. The glycerol then gets phosphorylated into triose phosphate, and it can enter aerobic respiration at the Krebs cycle to be used. Proteins can also be modified and used. They'd have to be hydrolyzed into amino acids, and then the amino acids would have to be hydrolyzed and the amine group removed, or deamination. The carbon compound that remains could then enter the Krebs cycle. So let's have a look at all of these stages, starting with glycolysis, because that's the first stage. And this actually happens in both aerobic and anaerobic respiration. No oxygen's needed in this stage, which is why it's in both types of respiration, and it happens in the cytoplasm. So there's 3 key stages in glycolysis. Phosphorylating glucose to form glucose phosphate, and that requires ATP. The production of triose phosphate, and the oxidation of triose phosphate to produce pyruvate with the net gain of ATP and reduced NAD. So NAD is one of the coenzymes used in respiration, and it has the same role as NADP did in photosynthesis. Do not confuse those though, it will cost marks. So the way that I always say to remember it is NADP, think P for photosynthesis. So here is the glycolysis reactions. We start with glucose, a 6-carbon compound, and we said that is phosphorylated to glucose phosphate. Phosphorylation means adding on phosphate groups, and that's why we have to use ATP. 2 molecules of ATP get hydrolyzed, and that means the inorganic phosphate removed from those 2 molecules can be added onto glucose, and that makes glucose phosphate. That is a more reactive molecule, and that causes it to split into 2 molecules of triose phosphate. That triose phosphate is then oxidized to make 2 molecules of pyruvate. And we can tell it must be an oxidation because the coenzyme NAD has gained a hydrogen from the triose phosphate. And if it's gained a hydrogen, that means it's a proton and an electron. So if triose phosphate has lost that proton and lost the electron, then it has been oxidized, and the NAD has been reduced because it gained that electron in the hydrogen. So you can call that NADH or reduced NAD. Now this oxidation reaction also produces 2 molecules of ATP. So this means from 1 glucose molecule, we get 2 pyruvates, 2 reduced NAD, and a net gain of 2 ATP molecules. And that's because although we've made 4 in total, we had to use 2 to start with. So it's a net gain of 2. Mark schemes are incredibly picky that you say net gain of ATP, even better if you can say a net gain of 2. So now we've got our pyruvate and those 2 molecules of reduced NAD. Those are going to be actively transported into the mitochondrial matrix to be used in the next stages. The ATP can be used in the cytoplasm for other reactions. The link reaction is the next step, and this is when pyruvate is going to be oxidized further into acetate. And again, we can tell it's an oxidation reaction because NAD has now formed reduced NAD. So NAD has been reduced, which means pyruvate must have been oxidized. 1 carbon has also been removed, so we've formed 1 carbon dioxide molecule. So that means we've gone from pyruvate, which is 3 carbons, to acetate, which is 2 carbons. Now acetate can't enter the Krebs cycle as it is, so it has to combine with a coenzyme called coenzyme A to make acetyl-CoA to enter the Krebs cycle. So that's a link reaction, and for every glucose molecule we end up with 2 acetyl-CoA, 2 carbon dioxide, and 2 reduced NAD. And the reason for that is for every glucose molecule, 2 pyruvate molecules were made in glycolysis. The Krebs cycle then, and this is actually just showing you the link reaction leading into the Krebs cycle. Acetyl-CoA is going to combine with a 4-carbon molecule. So we've got 4 plus 2, that makes a 6-carbon molecule. And once that has happened, coenzyme A is released and it can be reused in the link reaction. We then have a series of redox reactions where we go from having a 6-carbon molecule back to the 4-carbon molecule. And you don't need to know all of the intermediates, you just need to know that when you go from that 6-carbon molecule back to the 4, there's a series of redox reactions. And the carbon compounds are being oxidized, the coenzyme NAD and FAD are being reduced. And we get 3 reduced NAD being made and 1 reduced FAD. Because we're going from a 6-carbon to a 4-carbon, we've also lost 2 carbons to create 2 molecules of carbon dioxide. And that whole stage also releases 1 molecule of ATP. So the product per cycle is 3 reduced NAD, 1 reduced FAD, 1 ATP and 2 carbon dioxides, but per glucose molecule, which is really how we should do it, because in respiration, in aerobic respiration, we will have 1 glucose molecule entering, which means the cycle will happen twice because we get 2 pyruvate, therefore 2 acetyl-CoA. So that means from 1 glucose, we have 6 reduced NAD, 2 reduced FAD, 2 ATP, and 4 carbon dioxide molecules. So if we have a look at everything we've made so far then, we have got in total from glycolysis, link reaction, and Krebs cycle, 10 reduced NAD, because we had 6 made here, 2 made here, and 2 made here, taking into account how many times each of these processes occurs. We have 2 reduced FAD from the 2 lots of the Krebs cycle. We have a total of 4 ATP molecules so far. We had a net gain of 2 in glycolysis, and we had 2 from the Krebs cycle happening twice. And we have 6 carbon dioxide, which is waste, it's not useful, but we do have 6 carbon dioxide molecules. We had 2 being produced in the 2 lots of the link reaction, and 4 being produced in the 2 lots of the Krebs cycle. These types of questions are becoming more common, where you have to say how many of a particular molecule you have and identifying where it came from. So you do need to make sure you know all of this chemistry. So the final stage is oxidative phosphorylation. And this is when most of the ATP is made, because so far we've only made 4, a net gain of 4, which is not enough for metabolism. And this stage involves the transfer of electrons and the movement of those electrons along that inner mitochondrial membrane and the protons moving across. So in this one, first of all of those reduced coenzymes that we made are going to accumulate here in the mitochondrial matrix. The hydrogen is removed, and that is then split into protons and electrons. The electrons are picked up by these proteins and are passed down a series of electron carrier proteins embedded in that inner mitochondrial membrane, and this is the electron transport chain. As they move from protein to protein, they release a small amount of energy, which is used to pump the protons from the mitochondrial matrix into the intermembrane space, which is the gap between the inner membrane of the mitochondria and the outer membrane. That then creates an electrochemical gradient, meaning lots of this ion, the hydrogen ion, on one side. And as a result, the hydrogen ion can move down its concentration gradient through the enzyme ATP synthase, and that enables ATP synthase to phosphorylate ADP, meaning ADP plus PI, into ATP. So that is how we get lots of ATP being produced. And this is almost identical to that chemiosmosis that we saw in photosynthesis as well. Final thing then is the role of oxygen. And this is a really common key marking point. Oxygen is the final electron acceptor. What that means is oxygen will pick up the electrons at the end of the electron transfer chain. And it picks up those electrons, it also picks up the protons, and that then forms water. And it's really important that oxygen does this, because by picking up the electrons at the end of the chain, it means the electron transfer chain can continue, so protons continue to be pumped, and therefore ATP continues to be made. Anaerobic respiration is when there is no oxygen, and it only happens in the cytoplasm. The pyruvate that's produced in glycolysis is reduced instead of oxidized, and it will form ethanol and carbon dioxide if it's a plant or a microbe, or lactate or lactic acid in animals. And this is by the pyruvate being reduced because it gains the hydrogen from reduced NAD. That reoxidizes the reduced NAD into NAD so that it can be used in glycolysis again to make sure that at least glycolysis can still continue even though there's no oxygen, so that you get some ATP. So let's just have a look at that then. Here's glycolysis, as we just learnt, and in animals, lactate is made. So that pyruvate is reduced into lactate by gaining the hydrogen, which is a proton and an electron. That NAD that has been made or regenerated, reoxidized, can then be used in glycolysis again, so at least you still continue to get a net gain of ATP. Exactly the same in plants and microbes, but the product's different, ethanol and carbon dioxide. Next then is 3.5.3, energy and ecosystems. Now in any ecosystem, plants are going to be the producers in a food web. And that's because they produce their own organic compounds from photosynthesis, like we just saw. And between each trophic level, meaning the stage in the food web, energy is lost. And that's because between each stage, the organism will be respiring, so using up that glucose, and therefore there is less biomass available in the organism. And also there'll be energy lost through excretion as well. Everything that remains that is available for the next organism to consume is the biomass, and the amount of biomass remaining in the organism can be measured in terms of mass of carbon or dry mass of tissue per given area, but usually it's mass of carbon per given area. So to measure that dry biomass, if you wanted to know what the amount of biomass was, because it is the dry mass or the mass of carbon, you have to dehydrate it. And in order to dry out your sample to measure the biomass, you have to heat it in an oven at temperatures lower than 100 degrees C. Now, this has come up a few times recently, and students didn't get the mark unless they said heat. You can't just say put it in an oven. You have to specify heat it in an oven, and it has to be at a temperature lower than 100 degrees C, because anything above that will cause it to burn, and if it's burning or combusting, you're making carbon dioxide, and that is removing carbon. So heat it in an oven lower than 100 degrees C, and you need to constantly weigh while you're doing this drying, and then when the mass becomes constant, that means you now have just the dry mass remaining because no more water is evaporating, which is why the mass would have been decreasing. The chemical energy stored in dry biomass can also be measured or estimated using calorimetry. And this is done by burning the sample in a bomb calorimeter. And then the energy released from that combustion is used to heat a known volume of water. What you would do is measure the temperature increase, and that is then used to calculate the amount of energy that there must have been in the biomass, because that energy has been converted into heat energy, so it'd be the temperature increase. And that is measured in joules or kilograms per gram. Next is known about NPP and GPP. How productive an ecosystem is depends on the abiotic and biotic factors. So if there's plenty of favourable abiotic factors, water, a high light intensity, optimum temperature, then green plants will be able to photosynthesize at their maximum rate and therefore produce more organic substances such as the carbohydrates, and therefore it's more productive. And this can be quantified using GPP and NPP. GPP, which is gross primary production, that is the chemical energy stored in plant biomass in a given area or volume. So basically, it's the total energy resulting from photosynthesis. Net primary production, or NPP, that's the chemical energy stored in plant biomass after respiration losses have been removed. And that is basically what is available for the next organism in the food chain, because you've taken away the losses, the energy losses already. Now, if you are asked what either of those terms mean, always give the equation, because that is always worth a mark. The NPP, as I said, that is the energy left over after respiration has been removed, and therefore it's what there is available to make new biomass. Another equation you need to know is the net production of consumers in a food web. And to work out the net production of consumers, known as N in this formula, you would need to do I, which is the chemical energy stored in the ingested food, minus the energy lost. And animals can lose energy through feces and urine and respiration. So we can see we've got our energy input through the ingested food minus the energy lost through respiration and through feces and urine. The productivity refers to the rate at which biomass is produced. Primary productivity is the rate at which producers convert light energy into chemical energy or biomass via photosynthesis. Secondary productivity is the rate at which the consumers produce biomass by eating other organisms. And the rates of productivity are recorded using these units: kilojoules per hectare per year. So kilojoules is the unit for energy, but we do it per area and per year. And the reason we do it per area is so that we can standardize the results, taking into account that if you have a bigger area, you probably have more plants, so you would have more energy. And we do it per year to standardize the impact of the seasons. Because if you measured one area in summer and another in winter, summer would have a higher productivity because there's more plants photosynthesizing. Next is 3.5.4 nutrient cycles. Nutrients are recycled within natural ecosystems, and we see this in the nitrogen cycle and the phosphorus cycle for AQA. And microorganisms play a vital role in both of these cycles to recycle the elements phosphorus and nitrogen. Mycorrhizae are the fungal associations that you get between plant roots and beneficial fungus. And this is going to be useful because the fungi has these hyphae which can branch through the soil and increase surface area for water and mineral absorption. The mycorrhizae also act a bit like a sponge. They can really absorb and retain water. So that makes it quite drought resistant because that water and minerals is going to be shared with the fungus and the plant. So this is vital in the nutrient cycles because if there is a soil which is quite low in either nitrate ions or phosphate ions, having mycorrhizae present will increase the ability to absorb that for the plant. And this is a mutualistic relationship because the plant also provides a benefit to the fungus because plants photosynthesize and it can share some of those carbohydrates with the fungus. So let's look at the nitrogen cycle. The nitrogen cycle is a really important one because nitrogen is needed for making proteins, ATP and nucleic acids. And the challenge in this cycle is, although nitrogen is very abundant in the atmosphere, nitrogen gas has a triple bond between those two nitrogen atoms, and we do not have an enzyme that can break it. However, microorganisms do. So microorganisms are incredibly important in this cycle. There's 4 key stages: ammonification, nitrification, nitrogen fixation, and denitrification. All of these stages use microorganisms. So step 1 is nitrogen fixing or nitrogen fixation, which I've labelled on the diagram. This is when the nitrogen from the atmosphere is fixed by bacteria, which means bacteria break that triple bond between the N2, the 2 nitrogen atoms, and it fixes it, meaning converting it into a nitrogen-containing compound, which is ammonium ions. Now that can either be done by bacteria that are free living in the soil, so the ammonium ions are fixed into the soil, or there are some plants, such as leguminous plants, which are clover and beans, that have these nodules, which means lumps, on their roots. And within those root nodules, there are bacteria that can fix that nitrogen directly into the plant itself. Step 2 is nitrification. This is a 2-stage oxidation reaction. The first step is those ammonium ions are nitrified into nitrites by nitrifying bacteria. Nitrites are then oxidized further, again by nitrifying bacteria, into nitrates. And those nitrates can then be absorbed by the root hairs of the plant and be assimilated, meaning used in the plant and converted into proteins or DNA. And to get into animals, animals eat the plants. Ammonification links to the death and decomposition of animals and the decomposition of plants. But here is where you have to be really particular with your language to get the mark. You have to name the molecule that would be in the dead animal or plant or in the waste. You can't just say a dead animal or plant is broken down. So for example, proteins in a dead animal or proteins in a dead plant, urea, which is an example of the waste, or DNA in the dead animal or plant, you'd have to name it. Those are then decomposed by saprobionts. And saprobionts are bacteria and fungi that can digest dead matter or waste outside of a cell, so extracellularly, and that then reforms the ammonium ions. Now the last stage I've not actually labelled on, it's over here, nitrification. This is not a useful stage because This is when those nitrates could be converted back into nitrogen gas, and therefore you're losing nitrogen compounds that could be absorbed. This only happens if there are anaerobic conditions, and that would then mean that denitrifying bacteria that do this process would be able to thrive, and these ones that are all aerobic bacteria wouldn't. So this is why it's so important to keep soil aerated, so to plough it and toss the soil before you replant seeds, to make sure that there's plenty of oxygen so that you get the beneficial stages happening and not the denitrification. It's also why having waterlogged soils can be an issue, because the water displaces the air and then you'd have the denitrifying bacteria as well. Final note is a key tip for the exam, and that is making sure that you are saying nitrogen containing compound for any of these stages. We only say nitrogen when it is literally the element in the atmosphere. All of these other stages we have a nitrogen containing compound. So then we have the phosphorus cycle, and phosphorus is really important because it's used to make the phosphate group in DNA, RNA, ATP, and in a phospholipid to make the phospholipid bilayer. And this cycle is quite different to other ones because there isn't a stage where you have it as a gas in the atmosphere. And instead you mainly have it stored in phosphate ions in a mineral form in rocks. So here's the phosphorus cycle. If we start from phosphate ions in oceans and soils as a store, it's absorbed into the plant by active transport. That would then be converted into maybe DNA or phospholipid bilayer. Animals would consume that, and that's how it gets into animals. We would have some return to the oceans by decomposition of the plant and decomposition and excretion from animals. But we also have phosphate ions from waste and remains, such as bones and shells that don't get broken down as rapidly. And also guano. Guano is this bird pea, this white bird pea that you can see here, which is very rich in phosphorus. Now, those bones and shells that aren't going to be decomposed by the saprobionts will be broken down through erosion over time, and that returns the phosphate ions back to the ocean and soils. But we can also have rock formation happening. And this could be deposition or sedimentation. So those phosphate ions within the water or in the soil, over many, many years, heat and pressure, it creates those phosphorus sedimentary rocks. And to get that back into the water, again, it'd be erosion. Now, fertilizers, this is going to be something that you'd have to artificially add to get more phosphorus back into the soil. So here are our key stages. We've said the weathering of rocks, and that releases phosphate ions into soils and water. Plants absorb phosphate ions through their roots. Animals obtain phosphorus by eating plants or other animals. Excretion and death will return phosphorus to the soil or water. And decomposers, so those saprobionts, can break down dead organisms, releasing phosphate ions into the soil. Fertilizers are really, really important to replace nitrate and phosphate ions lost when either plants or animals are going to be harvested and removed from the cycle. Because if you're removing a plant or an animal from your cycle, then you're removing those mineral ions that are locked up in their cells, and also they're not there to decompose and return it back to the soil. So you always have to use fertilizers, and that can either be natural, so using manure, because we know from those two cycles that in feces there are phosphorus, phosphate ions and nitrogen containing compounds that can be broken down. Or you can use artificial fertilizers, which are inorganic chemicals, meaning no carbon, and there are pros and cons of these too. Natural fertilizers are cheaper. If you have animals on your farm already, it's free, it's just their manure. However, you don't know the exact mineral content, so you don't know how much nitrate or phosphate there is in there, and you can't control that. So artificial fertilizers, that is their main advantage. You can control the exact minerals and the proportions that you have. But you do have to pay for them, so they're going to be more expensive, and they're more harmful for the environment. And that's because inorganic substances are more soluble. Therefore, when it rains, they're going to dissolve in that rainwater, and they can then be washed away to water sources and have a negative impact on the environment. So the sorts of environmental impact, first of all, leaching, and that is when water-soluble compounds such as the nitrates or phosphates get washed away from the soil into rivers or ponds or lakes nearby. And if nitrogen fertilizers leach into waterways, that causes eutrophication. So eutrophication is when nitrates have leached from the fertilized field into the pond or lake, whatever water source it might be. And that causes the algae in the lake to grow much more rapidly. That creates a blanket of algae across the surface, and that blocks sunlight from reaching the lower levels in the water and any lower down plants. So if those plants lower down aren't photosynthesizing, they die. Bacteria within the water will then decompose those dead plants, but in order for bacteria to do that, they have to respire more, which means they're going to be using up oxygen. So using up all that oxygen eventually means there's not enough oxygen dissolved in the water for all of the other aquatic organisms, such as fish. And if fish don't have oxygen, they can't respire and they die as well. So that takes us to the end of topic 5. Hopefully you found it helpful. If you did, don't forget you can check out my A-level notes and flashcards for mark scheme specific answers. That's it for today. Hope you found it helpful and I'll see you in a video very soon.